agents. So till the time we have studied what are the viruses, let us say HIV, human immunodeficiency virus, which can cause the AIDS. And we have seen other examples such as coronaviruses, then the flu, and influenza, swine flu. So these are all the viruses which can cause lot of disturbance in our routine. Okay, so we know that air is one of the common medium for our survival. But if air itself is polluted, then under these conditions, it is challenge for us to protect ourselves from the viruses. Okay, so these kinds of the viruses, we have seen their genetic machinery. That means these are the viruses which are having certain genetic material, protein code, and envelope. So this is the kind of schematic representation of their composition. Okay, so these are the viruses which generally they have either RNA or DNA in their genetic machinery or their genetic composition. This is basically having either RNA or DNA. So the question is with us, uh, how these viruses are So in the last documentary, basically we have seen one of the video where all these viruses which are present in our body, how they can multiply from one to many. So and depending upon their transcription and translation process, several types of the enzymes are involved in multiplication or duplication of such virus particle. So single virus particle is called as virion, or we can pronounce it virion. And the many viruses are there, then it is called as virus. Just like if we have only single bacterium, then it is called as bacterium. But if there are more than one bacteria, so they are pronounced as bacteria. So singular word is the bacterium and plural is like bacteria. So similarly for virus, a single virus particle is termed as virion. But if we have more than virus particles, so they are termed as virus. Okay, so viral particle is consist of, these are the things which are present on the your screen, and then these are the various ways to classify the antiviral drugs. So depending upon the cellular machinery or the kind of compositions of all these viruses, these virus particles are classified according to or antiviral drugs are classified depending upon their type of the mechanism or their according to their mode of the action. Okay, so here the first is this, which are the various viral attachments and entry blockers. So basically they are enfluvertide and dofenisonazole, pembrolizumab. So this is a monoclonal antibodies, pembrolizumab, which is useful in respiratory syncytial virus. So that is viral attachment and entry. Then uncoating inhibitors, we will also visit these uncoating inhibitors, which are going to inhibit the uncoating of the viruses. Basically, they are amantadine and remantadine, which can cause influenza. So amantadine and remantadine are the drugs which are used in influenza. Then viral release blockers. So neuraminidase inhibitors, they are useful in these conditions. This is again they are useful in influenza. Then basically oseltamivir and zanamivir, if you might have heard the name of these drugs. So these are the drugs which are mainly inhibiting this influenza in neuraminidase enzyme. Just like if we call it as H1N1 swine flu. So H1N1 swine flu, So basically that is hyaluronidase, neuraminidase 1, hyaluronidase 1, neuraminidase 1. So that is this enzyme, neuraminidase, which can inhibit the release of the viruses from the cells. Means one cell can multiply to many other cells. Then nucleic acid synthesis inhibitors, depending upon the types of their structure, these uh, antiviral drugs are classified as either nucleoside reverse transcriptase inhibitors or non-nucleoside reverse transcriptase inhibitors. They are also called as NRTI, the abbreviation of nucleoside reverse transcriptase. And non-nucleoside reverse transcriptase, basically they are termed as NNRTIs. So they are majorly used in HIV or some other kinds of infections. Okay, then protein processing, which are basically protease inhibitors and HIV inhibitors. So they are useful in the HIV. So protein processing inhibitors are basically all the kinds of protease inhibitors. So these are the drugs which are inhibiting the viruses and their multiplications. So maybe that these are the only applicable for some of the enzymes. I mean to say, some of them are used in HIV, influenza, or likewise. But we know that some of the protease drugs are also useful in certain COVID-19, just like remdesivir, which is RNA-dependent RNA polymerase inhibitor, which is one of the enzyme which is protease, RNA-dependent RNA polymerase. So that is polymerase enzyme, and that is useful in this treatment of COVID-19, coronavirus disease 2019. So there are several other drugs which are used in such way, and they have their different mechanism or mode of the actions. So what the way as of now people are doing, people are generally taking these molecules and they are working against their targets, and they are doing a lot of virtual screening and mediated through computational chemistry or computer-aided drug design in search of identification of new inhibitors of COVID-19. Fine. So this is the way that research against COVID-19 is progressing. But our discussion is on the viral machinery, the viral cells. Okay. So, friends, now next there is several other uncoating inhibitors we have to learn. So among these inhibitors, basically amantadine and rimantadine, these are the drugs which are useful in uncoating inhibitors. So what are these? Basically, they are hydrophobic amines. You will see then come to know that they are having ring structures. I mean to say aliphatic, alicyclic ring structures into their composition. And they are hydrophobic amines, just like your methylamine, ethylamine, propylamine, butylamines. They are the hydrophobic amines, or we can also call it them as weak kind of organic bases, which are having activity against influenza A. So there are several other types of the influenza A, influenza B, and so many more. Okay, virologists can define more clearly that what is the difference between influenza A and influenza B. Okay, specificity stands for, they are also called as the blocker of M2 protein, so M2 protein inhibitor. And here is a mechanism that somehow it is shown that viral particle will some enter and how the way it will undergo uncoating. Uncoating is the, I mean to say, what are the genetic machineries, etc., which is present in such cells. It will come out and will show somehow kind of, which will release into the cellular organism. Okay. So what are the drugs? We will not go much more depth into this mechanism of action of this N2 inhibitors, but just for the sake of simplicity, remember that amantadine and rimantadine, these are the drugs which are going to inhibit the N2 channels. They are useful in influenza against the type A influenza viruses. So friends, this is the structure of this amantadine. Now you can see how it is very much closely related to Basically, this is one cyclohexane ring which is directly attached with NH2. Okay, so that is primary amine, aliphatic amine. Just like after methylamine, so it is CH3NH2 and ethylamine. So ethylamine is primary amine. So here is also, this is a primary aromatic amine. And this is a cyclohexane ring which is connected with another form of cyclohexane ring. So it is a bicyclic compound as of now, if I say. But not only that, these two carbons are connected with this methylene single bridge, or single carbon is the one which is attached between this. So in this structure, if we try to identify the cyclic rings, can you identify the number of cyclic rings present in this structure? Options are 1, 2, 3, or 4. So how many cyclic rings are present in the structure? Then definitely, let me tell you the answer. Answer is, this is a structure which possesses 3 cyclic rings. Number 1 is this hexagonal cyclic ring. Second is this chair conformation, and this is third ring. And this, I mean to say, this chair conformation is a third, and this ring is again fourth. Sorry, so it is not 3 there are 4 cyclic rings. There are 4 cycles. There are 4 rings present in this structure of amine. Okay, and other name of the same is 1-amino admentane. So actually, this is a bicyclic, I mean to say a cyclic cage. Right? It is called cage. Cage means just a cage for a parrot, cage for a sparrow, or something other kinds of birds. Okay, so this is also kind of a cage structure. 4 cyclic rings in a one drug molecule. Okay, so basically this is going to inhibit uncoating of viral RNA to infect within the host cells, so that it can prevent the replication of such viruses. Okay, so I hope the structure is clear and very much easy to draw. You draw a cyclohexane ring, draw a chair conformation, and draw a single methylene bridge in between the top carbon and the bottom carbon. You'll be able to make the structure of adamantane. Okay, and just simply draw a primary aromatic amine NH2 on the top of this ring cage. So that will allow you people to identify your, or to draw the structure molecule of amantadine. Yeah, so this is the similar derivative of amantadine. It is rimantadine. Rimantadine is basically similar analog. Just the only the difference is but it possesses this, I mean to say, CH3CHNH2, CHNH2. So that is present in the structure of rimantadine. And this rimantadine is not only antiviral drug, it is also used in Alzheimer's disease. So this is a drug which is useful in dementia, in senile dementia, where the people will forget the loss of memory at a longer age, at the higher age. And under this condition, this rimantadine is one of the drug which is used as anti-Alzheimer drug. So that is marketed, and it is also having its antiviral effect. Likewise, this is a kind of what is a purpose that is called as repurposing of the drugs. This is a drug which is antiviral, although it is used in Alzheimer's disease. You know several other examples of such repurposed drugs. Repurpose is, I mean to say, we are trying to make more than one purpose of these molecules. It's called repurpose. Already the molecule is used for certain purpose, but that is all a repurpose. Just like it is like for a teacher who teaches you medicinal chemistry also teaches you organic chemistry, and sometimes is also eligible to teach you jurisprudence, or also may teach you biostatistics, or may teach you pharmacology, pharmacotherapeutics. A same teacher who is teaching you multiple subjects. So similar, if a teacher can, a human being can teach multiple subjects, And why not a drug molecule used for one disease, the same molecule cannot be used for another disease? It is also possible. So this way, these drugs are repurposed. Aspirin, right? You know the structure of this molecule. Benzene banao, COCH3, COOH. Both are at 1,2 position of benzene, right? So this is the structure of aspirin. And aspirin में क्या है? It is going to be, it was an NSAID, non-steroidal anti-inflammatory drug having antipyretic effect, then anti-inflammatory effect, and analgesic. So these are the three actions that are present with aspirin. Now we know that this is the same drug is also used in blood thinning, some cardiac disorders. This it can cause blood thinning also, so that's why it is used in cardiac disorders, the disorders related to the hearts. So that is what called as repurposing of drugs. In COVID-19, this terminology is repeatedly used in field. Repurposing of the medicine. It was already used for the medicine. This is a way that it will, because what will happen, drug discovery is a big process. It will require 13 billion US dollar to bring a single molecule into the market. इतने पैसे मेरे और आपके पास नहीं है, या आपकी फैमिली के पास, किसी के पास भी नहीं है. 13 billion, not Indian national rupees, they are US dollar. 13 billion US dollar. And maybe we require a lot of money to make a molecule into the market, bring a molecule into the market. So under these conditions, the question is how to proceed for drug discovery. Such a highly costly drug discovery process, and it requires a lot of time, 13 to 15 years, or sometimes even more than 10 years to bring a molecule into the market, right? So the repurposing of the medicine is perfect example for आज COVID-19 में what the people are doing. There is no medicine for COVID-19. Remdesivir is basically some other was used in some other kinds of the viruses. That is given. Chloroquine or hydroxychloroquine, they were used as anti-malarial agents. They have been given as a prophylactic to the health workers. Okay? So this is a kind of, I mean to say, prophylaxis or a repurpose of the medicine. Or there are a lot of other medicines which are used. Some protease inhibitors. They are used in some other kinds of viral disorders. They are now also being used for this purpose against COVID-19. Okay, so this is basically synthesis of amantadine. This is a cyclic cage, which is called as adamantane. If it is brominated, so it is converted into 1-bromoadamantane. It is reacted with acetonitrile in the presence of acidic conditions, so that it will form this acetamide derivative, and upon hydrolysis of this carboxamide we can have amantadine. A simple straightforward synthesis. We will take adamantane, brominate it, acetylate it, and perform the hydrolysis. So this way we can have constitutions of synthesis of this amantadine. This is the synthesis of remantadine. We shall take this acetyl derivative of adamantane, react it with hydroxylamine. We will also use the same reagent in our laboratory, hydroxylamine hydrochloride, and it will form the oxime. This is hydroxyl derivative of this, which is also called as oxime. Okay, and reduce it with lithium aluminum hydride as a reducing agent. So that is a universal reducing agent, will reduce all the functional groups. In this case, it is imine. So the reduction of imine by lithium aluminum hydride can give amine. This way we can synthesize rimonidine, another molecule or entity we drug. Okay, then it comes to the entry inhibitors. So these are some other entry inhibitors which are basically whenever virus particle enter into the host cells, then there will be dissolution of the viral code and the genetic machinery will be released into the host cells. So can we do somehow that we can prevent the entry of such virus into the cells? So the preference, these are some of the drugs, all as Viroc, enfluvertide, and docosanol. These are the new drugs. Viroc, which is a new class of antiretroviral agents. Uh, you might be knowing that HIV viruses are also called as retroviruses because their genetic composition is having, I mean to say, their genetic composition is having RNA as their genetic machinery. So that's why they are having, they are also used for this purpose, retroviruses. Okay. So antiretroviral agent which is used in HIV, that is this molecule. Again, this is a structure of the same. And here this is a benzene ring, cyclohexane ring, this is a carboxamide, and here it is triazole ring. 5-membered ring having 3 nitrogen in its structure. That is called 1,3,4 triazole. First position पे भी nitrogen है, third पे भी है, fourth पे भी है. 1,3,4 triazole. So here it is what's so called as kind of this ring structure. Again, this is a 5-membered ring pyrrolidine and it is attached with a 6-membered ring. Not 1, 2, 3, yeah, it is 6-membered ring then. That is I mean to say piperidine, and this is again a bridge aid. It is the nitrogen is the atom which is present in this case as a bridge aid. I mean to say BRIDG. So it is called as bridgehead because nitrogen is connecting, is present at the attachment of these two cyclic rings. So that's why this terminology has came into the existence. Bridgehead, where a single nitrogen atom is present between the two cyclic rings. So this is going to inhibit the HIV-1 viruses and useful in immunodeficiency syndrome, acquired immunodeficiency syndrome. This is the synthesis, although that is not of our more importance to us. This molecule we have to study, although its structure is difficult to remember, but it is a new class of drug, enfluvertide, which is going to inhibit the inhibition of various membrane of viral viruses, right? And it has shown significantly the efficacy in combination with other antiviral drugs in early HIV detection. And you can see the structure of this drug. This is again made up of some peptides, a big peptide, although it is not a cyclic one because it is an acyclic one. So this is a tryptophan, again tryptophan, this is a serine, this is tryptophan, this may be lysine, and this may be this is valine. Here this amino acid is again valine, and this is again valine. Here it is glutamic acid, again glutamic acid. This can be CONH, carbamic acid is there, and these are the several other carboxylic acid and carboxamides are present into their structure. This is histidine, imidazole containing amino acid. So several amino acid structures are present in this peptide drug. I will call this enfluvertide as a peptide drug because its structure is composed of lot of peptides. Okay. So this is basically a peptidal drug used as an enfluvertide, right? So next class of the drug is protease inhibitors. What are the protease? Basically, protease are the enzymes which are present in the genetic machinery, and they are useful in conversion of inactive proteins into the active proteins. Okay, so it is a part of viral proteins. Okay, so when a particular virus cells is there, so it is having so many compositions, RNA, HI, proteins, and protease enzymes. So if you want to inhibit such enzymes, then we can take the help of protease inhibitors. And remember, whatsoever the protease inhibitors are there, the term in this you must be aware about the prefix and suffix. Prefix is always at the beginning of a word. Suffix is at the end of the word. So here, all these viruses, which are, I mean to say, drug molecules, which are protease inhibitors. So they are having always suffix vir. It is not just like that वीर और brave person. वो आप सलमान खान का एक मूवी आपने देखा होगा जिसमें उसका नाम वीर है. I don't remember the name of that movie. Okay, but yes, here Veer is not that Salman Khan. Here Veer is protease inhibitor. So what are these? All the protease inhibitors. Examples are sakinavir, ritonavir, lopinavir, indinavir, and nalfinavir. Okay, although you may not be able to see some of the structures more correctly, but believe me, these all are viral, antiviral protease inhibitors. Sakinavir, which is having phenolene ring in its structure. This is a heterocyclic phenolene ring. उसके नाम में QUI है. So that QUI is nothing but वो आपका जो QUI है, that is a kind of heterocyclic ring named as quinoline. तो ये सकीनावीर का नाम आपको you can easily remember. It is a protease inhibitor and possess this phenolene ring. Okay, this way medicinal chemistry becomes easy. If you go on remembering each and every drug molecule और रट्टा मारने की कोशिश करोगे, you will try to mug up the heterocyclic rings present in the way. You will not be able to remember it for longer period of time. एक दो साल बाद आप थोड़ा भूल जाओगे, पर आप ऐसे नॉलेज रखोगे. सकीनावीर, so Vir stands for it is protease inhibitor, QUI stands for it is a phenolene drug, और ऐसे से आप कुछ और भी बना सकते हो. That way you can remember. Ritonavir, this is a drug which possesses thiazole. This is a 5-membered ring having nitrogen and sulfur in its structure, so that is called as thiazole. So this thiazole is the ring. This is the correct spelling. Thiazole, that is the heterocyclic ring which is present in ritonavir. So ritonavir may be on a body, you can highlight T is there something. So there are thiazole ring you can remember. This is lopinavir, and it is all the only heterocyclic ring present is this 1,2,1,3-diaza cyclohexanon, right? 1,3-diaza cyclohexanon, that is present in lopinavir. Indinavir, Indi is there. So Indi is basically for your 6-5-membered heterocyclic ring, that is indol. Okay, so, sorry, not indol, but this is 6-5-membered ring. It is termed as indene. Okay, so yeah, this cyclic ring is called as indene. Whenever 6-membered nitrogen 6-membered ring is attached with a 5-membered ring, but there is no nitrogen into that, so it is called indene. But if nitrogen is present, so it is called indole. Okay. So in this case, there is no not a heterocyclic ring is indole, but it is called indene. So indene se naam aaya hai indinavir. Okay, nelfinavir, definitely whenever you say nelfinavir, so here this is a bicyclic ring is present, and it is there. So phenyl ring is also present. There are two phenyl rings, and a tertiary butyl moiety is also present in the structure of this. And hopefully, the structures may be visible to you by this way. That these are the drug molecules having their somehow effect is the viral viruses. Okay, let us go back to our this. This is the synthesis of Saktinave, but we are not interested into that. Just we are going to learn the classification. Then more important type of the enzyme which is widely used in antiviral medicinal chemistry, that is nucleic acid inhibitors. Okay, and they are nothing but आपका जो reverse transcriptase enzyme, and so all they are the part of this. So either they are going to act against polymerase enzyme or they are going to inhibit this reverse transcriptase. And that is required for nucleic acid synthesis. Nucleic acid मतलब और कोई नहीं, but they are RNA and DNA. If they have ribonucleic acid, then it is RNA. If they don't have it, it is called DNA. Deoxyribonucleic acid. Okay, so friends, this is the way that these nucleic acid inhibitors are there, and these are their structures. Again, nucleic acid inhibitors are having their names as weed. So this may be weed, and so whenever we have protease inhibitors or RNA dependent RNA polymerase or reverse transcriptase enzyme is there. In that condition, this weed terminology is used. Acyclovir. Oh, this is the structure of acyclovir, and basically that is this adenosine derivative. So this ring which is present in the structure, that is called adenosine ring. 6'5" membered ring which is connected with purine type of the base. Purine base which is present in the DNA that is having 6'5" membered ring position. If you are not aware about this, then go back to your simple biochemistry by Satyanarayan and refer that knowledge of purine base or purine base. And if you cannot do that, then simply do the Google search. You will come to know what are the various nitrogen bases and how their structures are there. You can easily remember their structure. Okay, so 6'5" membered these are the bases which are basically useful as kind of this nitrogenous bases. Okay, in this case, let us say for example, purine base, they are adenine and guanine, and pyrimidine base, they are thiamine, uracil, and cytosine. Okay, so these are the three pyrimidine types of the base. So pencyclovir, that is basically kind of again I mean to say structure is more similar with that of this gencyclovir. Only the difference is here CH2 is replaced with oxygen. So that is oxa analog of pencyclovir and name as gencyclovir. A simple, all these are cyclovir. They belongs to adenosine derivative. Another adenosine derivatives, that is valcyclovir, and valcyclovir is nothing but that is this amino acid present. Valine amino acid. So valine is the new amino acid which is present in the structure of this and having composition of this. अब आपको कैसे पता लगेगा कि वो valine है, क्योंकि आप you can see CH3, CH and CH3. So it is a V shape. I mean to say isopropyl. That isopropyl V shape. So that way you can remember. Let us say it is valine amino acid. Okay, so valcyclovir is nothing but this is a drug which is a prodrug of acyclovir. I mean this. So there is what is the difference between this molecule and this? Only the difference is यहाँ का जो ऑक्सीजन हाइड्रोजन का जो हाइड्रोक्साइल ग्रुप है उसको रिएक्ट किया है COOH of valine. तो एस्टर बॉन्ड बनाया है. So that way, this is an ester prodrug. So naturally, being an prodrug, having an ester bond, it is going to hydrolyze by the another enzyme. The name of this enzyme is esterase. Esterase is the enzyme which is going to hydrolyze this bond. Once this is hydrolyzed, it will be your easy. Okay, we know what are the prodrugs. We visited there several lectures on prodrugs and several therapies having the for prodrugs. So that we have already visited in our previous lectures. Okay, so these are the I mean to say several inhibitors of viral DNA polymerase. Acyclovir, penciclovir, encyclovir, and right. So let us visit these molecules one by one. So we shall spend some quantitative time on each and every of this reverse transcriptase inhibitors, and all are nucleoside because these are the nucleosides. So let me go back to bring you people go back to what is the meaning of several nucleosides to present something just for I will try to make you people memorize something for the nitrogenous base. Okay, so let us go to that. Hope the screen shall be visible to you people very soon, but simply look at here. This is a nitrogenous base having purines and pyrimidines. Purines are having adenine and guanine. So they both of them are present in DNA as well as RNA. So here this is NH2, and but in this case NH2 is present in between these two nitrogen, which has been unsubstituted in adenine. Okay. Yeah, sorry. So I had made a mistake. It is not any adenine analog. It's encyclovir, valcyclovir, penciclovir are guanine analog. So they are guanosines. उसमें कार्बोनाइल ऑक्सीजन भी है और NH2 भी है. So they are guanine analogs. एडेनिन में खाली NH2 ही होता है. यहाँ पे डबल बॉन्ड वो नहीं होता है. They are present in DNA. They are present in RNA as well. These are the three pyrimidine bases. The monocyclic नाइट्रोजीनियस बेस उनको नाइट्रोजीनियस इसलिए बोलते हैं बिकॉज वो जो साइक्लिक रिंग है ना उसमें नाइट्रोजन जैसे ये दोनों साइक्लिक रिंग में देयर आर 4 नाइट्रोजन्स. In this case there are 2 nitrogens in each of this cytosine, thymine and uracil. Thymine और uracil में फर्क इतना ही है कि in this position there is methyl in thymine. Thymine का जो सिंगल लेटर है that is T. And in uracil there is no methyl. बस तो इतना ही फर्क है आपका डीएनए और बैक्टीरिया की डीएनए में. आपका डीएनए और रिट्रोवायरसिस का डीएनए में. Only this methyl. Atomic 1 carbon 3 hydrogens CH3 or atomic weight is 15. Okay, only the difference is this thymine versus uracil. Because as a Homo sapiens, we people have DNA, but somehow prokaryotes will have this with uracil in the DNA. I mean to say the genetic machinery. So all these are the structures which are basically called as nitrogenous bases. Purine, pyrimidine, adenine, guanosine, cytosine, uracil. They are nitrogenous bases. If you want to learn this in depth of the concepts, then go back to and library, or you can also refer to biochemistry by Satyanarayan, where you will identify what are the nitrogenous. So these are the drugs. We are having nucleosides such as guanosine analogs in their structures, and hence they are termed as nucleoside reverse transcriptase inhibitors. Okay, NRTIs. Fine. So guanosine analog, yes. So here it is the mistake which I have told you previously, that is now corrected. It is a guanosine analog because guanosine will have carbonyl oxygen and this primary amine. Yes. So this is a drug which is used in herpes simplex virus type 1 and type 2 and varicella zoster viruses such as chickenpox and shingles. You might have heard another disease of the name chikungunya. So chikungunya में क्या होता है कि their joints in between these legs or in between the hands they get stuff. They are not able to move properly. So if movement cannot occur, then patient will be sitting or sleeping at a particular one side. Voluntary movements are completely off. Voluntary muscles of the body will not work. Okay, and that's why. So under this condition, some of these antiviral drugs can be used. Just I remember chicken pox, तो मुझे चिकनगुनिया भी याद आ गया. So it is just like chicken. People will have some abruptions, tiny abruptions on the skin, and chickenpox is the name. Right? The vaccines are also available for the chickenpox, and maybe they can be given to prevent themselves. We are also aware about this. And the person who discovered this medicine for uh, this pox virus is Edward Jenner. Then આપણે ગુજરાતીમાં કહીએ ને શીતળા. So that vaccine has been discovered by him. So vaccination is also one of the good option to prevent yourself from the several other diseases. BCG vaccine is for tuberculosis, Bacillus calmette guerin. So that is for prevention of TB. If the small infant is there, so within a month, within a week of the birth, either on left, mostly it is given on the left arm, where a small baby is given and injected this BCG vaccine. But people say that BCG vaccine is also not effective to prevent the occurrence of tuberculosis. Okay. So that's why this I just came to that concept of vaccination, but that is again not of our syllabus, so we will not touch much more on this. Right. So this is basically how the way it is going to act. Acyclovir is metabolized by the kinase enzyme to monophosphate, and again kinase enzyme diphosphate, then again it will metabolize to triphosphate, and this triphosphate is going to inhibit this DNA, going to inhibit the reverse transcriptase enzyme. So as such, acyclovir is inactive unless and until it gets converted into triphosphate enzyme mediated through this enzymatic actions of kinase. Okay, so that way this acyclovir is. So we will restrict ourselves up to this, and we shall start our sessions from this onwards in coming lectures. So thank you everyone. Thank you so much.